I'm going to talk about the cellular level. So what is a cell? A cell is the basic unit of life, meaning that they have organization, the ability to reproduce and grow, use energy, respond to stimuli, and most importantly, maintain homeostasis. Also, many variations of cells within the human body that perform different functions. The main parts of a cell that I would like to talk about, which is the membrane, the cytoplasm, which has the organelles, and the nucleus. Right, so on the outside of the cell, it's going to be a cell membrane, which is composed of a phospholipid bilayer consisting of phosphate molecules on the outside and inside of the membrane, and a lipid layer in between. The primary function of a cell membrane is protection from external threats and the fusion of vital substances. Right, so on the outside of the cell membrane, it's going to be the glycoprotein and glycolipids. They serve as identifiers of cells. In between the phospholipid bilayer are going to be cholesterol molecules. Pretty much they help stabilize the phospholipid bilayer. And the more cholesterol molecules you have, the more stiff it's going to be. The less cholesterol molecules you have, the more fluid it's going to be. So other features of the cell membrane are going to be peripheral membrane proteins, which is going to be on the outside of the cell membrane, integral membrane proteins, which will be inside the membrane itself, and channel proteins. And that little swirly thing inside, in the middle of the cell membrane is going to be an alpha helix protein. Pretty much they serve to transfer certain molecules and or nutrients across the cell membrane itself. All right, so this is a phospholipid molecule. One unique feature about this is as empathetic, meaning that it's both hydrophilic and hydrophobic. And as you can see on the top with the phosphate head, it's going to be hydrophilic. And on the bottom here with the fatty tails, it's going to be hydrophobic. It's going to be the cytoplasm. Pretty much is going to be the area between the enclosing plasma membrane of the cell and the nucleus. Pretty much the cytoplasm contains cytosol, which is going to be the fluid portion of the cell, and the organelles that maintain homeostasis. And the cytoplasm are going to be organelles and other cellular structures such as cytoskeleton. I'll discuss that later on though. First, the organelles are pretty much are structures in the cytosol to maintain homeostasis within the cell and within the organism itself. Just think of them as tiny organs, and each individual organelle has a unique function to it. And there's also two categories for organelle, which is going to be membranous and non-membranous. Membranous means it has its own cell membrane, and membranous doesn't have a cell membrane. And pretty much all the organelles are going to be membranous except for the centrosome and ribosomes. Alright, as you can see, there's many organelles in the cell itself. So what we're going to do is we're going to start inwards and move outwards towards the out part of the cell. Alright, in the center on the left-hand side is going to be the nucleus. It's an organelle, but it's unique, meaning that it's unique because it carries the DNA for the human body, and it's also membranous, like I mentioned beforehand. On the outside, there's going to be a nuclear envelope with nuclear pores. The nuclear pores is where the ribosomes come out of, and within that cell membrane, it's going to be kind of its own like a cytoplasm, which is called caroplasm, and that houses all the chromatin, which is going to be your DNA. And the very center of the nucleus is going to be your nucleolus. Pretty much that is the main part for ribosome production. Right, and outside the nucleus is going to be the endoplasmic reticulum. There's two types. There's rough endoplasmic reticulum and smooth endoplasmic reticulum. Pretty much the main difference is that rough endoplasmic reticulum has bound ribosomes on the outer edges, and the rough ER pretty much serves to synthesize proteins from the ribosomes, and the smooth endoplasmic reticulum synthesize lipids and steroids, and also works to detoxify the cell In the bottom left of the cell, it's going to be this is a Golgi apparatus. Pretty much they're responsible for processing protein from the rough ER and packaging them into vesicles and send them to either to the cell membrane, the cytosol, or they get secreted outside the cell. Pretty much think of them as like a mail room, organizing and sending out packages for the cell itself. And we also have peroxisomes as well, would help get rid of toxic waste substances within the cell. And we also have lysosomes as well, which helps recycle waste and convert it back into building material for the cell. Centrosome in the cell, which is pretty much a bundle of microtubules, and they play a key role in cell division. And pretty much they remain dormant in the interphase cycle of the cell. We also have ribosomes in the cytosol itself. They're pretty much copies of DNA that come from the nuclear pores of the nucleus itself, and it pretty much helps in protein synthesis. Probably one of the most important organelles, besides the nucleus, even though they're all important in their own way, is mitochondria. Pretty much just a power supply of the cell. Their main function is to produce energy through cellular respiration, and it has its own membrane, its own matrix, and is also double membraned. One last thing about the mitochondria is that it has its own DNA separate from the nucleus itself. I mentioned before, it has its own cytoplasm as well. Pretty much it's specialized to break down large glucose molecules into smaller ATP molecules for cellular use. Important cellular structure is going to be the cytoskeleton. They're pretty much like the skeletons in our body, but in microscopic view, smaller essentially. Their main function is to provide support, transportation of nutrients, and locomotion. And they're they're composed of actin and lipid molecules. And there's also three types of cytoskeletons. The first one on the left, it's going to be microtubules. Main components of the centrosome, and they also set up pathways to send different nutrients across the cell. The diameter of the microtubule is going to be 25 nanometers as well. And the next one is going to be the microfilament, which is going to be 7 nanometers in diameter. They're mainly used for movement inside the cell, and one of their main functions is the physical separation during the cytokinesis period of mitosis. All right, the last one on the very right-hand side, is going to be the intermediate filament. It's going to be at least between 8 to 12 nanometers, and serves as structural support to resist mechanical stress for the cell. Like all things, every living organism goes through a cycle. It's no different from a cell. Pretty much there's two parts of the cycle, which is going to be the interphase, which is G1 through G2, and the mitosis phase. The cell spends most of its time during the interphase period, where it pretty much just grows and maintains homeostasis. So in the beginning of the cell cycle, it's going to be the G1 phase of the interphase. Pretty much just maintain homeostasis, just grows normally. And then as you get to the S phase, pretty much all the DNA within the nucleus will be copied. And then in G2, during the last part of the interphase, it would double in size right before the mitosis phase takes place. And when the mitosis phase occurs is when it divides into two separate daughter cells, and then it restarts the whole cycle. All right, during the mitosis phase is when the two daughter cells are made from the previous cell. The first part of it is going to be the prophase, is when the chromosomes condense and become visible. The prometaphase is when the nuclear envelope on the for the nucleus breaks down, and the centrosomes arrive at each poles and begin to reach out to the chromosomes. And during the metaphase, third phase, is when all the chromosomes are going to meet up in the middle of the cell, and the centrosomes will hook up to the middle of the chromosomes at the kinetochores. And during the anaphase is when the chromosomes get to pull, each side of the chromosomes are get pulled apart to one side of the pole. And then during the telophase is when the nuclear envelope reforms, and then the chromosomes begin to decondense as well. And the cytokinesis stage is when the cleavage begins from the actual microfilaments, begin to separate the cells into two daughter cells.